So today I'll be mainly talking about AMR or antimicrobial resistance, mainly focusing on the environment. I'm pretty sure you'd be already knowing what AMR is, but anyways, I'll just give a brief intro before we move on. So AMR or antimicrobial resistance is currently one of the major global health issues, and it's like topped like among the top 10 global health priority issues currently by WHO. And there has been a rising trend in AMR over the past few years. So, what is AMR? AMR or antimicrobial resistance is the ability of the microbes, like which includes bacteria, virus, and fungi. When these microbes they change over time and become resistant to medicines, which are commonly used to treat infections caused by these microbes. So that's what AMR is. So this makes the infections difficult to treat, leading to impossible treatment of deadly infections, causing even death or mortality of the patients. So who is at risk? We all are equally like at risk because it can affect any of us. So no one is free from AMR. And however, the most vulnerable categories are the young kids, that is the newborn babies, all the elderly, all the people with like weak immune systems, because they tend to catch the infections faster. So if they are getting an infection due to an antibiotic resistant bug, that's a serious issue which can lead to like a treatment failure and even death of that patient. And now like we are now already like we are now in a severe issue, and we have to act as soon as possible before we step into an era that is a post-antibiotic era where the world will be without any effective antibiotics. So what happens is that if we are moving on to a post-antibiotic era, even the common infections will be complicated to treat, and the treatment cost will increase, and it may actually become impossible task to even treat an infection. So we hope we don't like step into that era. So it's the right time. Each of us we have an like duty to act to tackle this issue. Now, when I talk about AMR in today's talk, I'll be mainly focusing on antibiotic resistance because I mainly work on with bacteria and antibiotic resistance. So, what is antibiotic resistance? Antibiotic resistance happens when bacteria change over time and become resistant to antibiotics, which are commonly used to treat them. And now, what are the causes of antibiotic resistance? We can look into that one by one. First of all, misuse and overuse of antibiotics. So, how can that contribute to antibiotic resistance? So, for instance, if you're getting infected, like I would say, like you just got a common cold or flu, and you know flu is caused by like flu virus. But if the doctor prescribes you an antibiotic for that, that is a misuse because antibiotics are not going to work against virus because it is used to treat bacteria. So there's a misuse or overprescription that's happening there, and that shouldn't be done. And the next one is like patients or like misuse. For instance, even like we tend to buy even for like if you're getting a fever, just like that you go like if you have access to like over the counter antibiotics, you just go to the pharmacist and you tend to buy some random antibiotics and take it. So that's the misuse there. That should not be happening. And then patients not finishing their course. So suppose you got infected with a bacterial infection, and then a doctor prescribes you an antibiotic, and he says you it's a 7-day course. But by the third day, you feel better. So you think, okay, now I can just stop taking the antibiotic. So the patient, if the patient is not finishing the treatment or not finishing the course, what happens is that there's a tendency this bacteria like it's not like all dead and it will recur. So an infection will come again, and then again you'll have to take the same antibiotic again. So again, that's an issue. Now coming to similar to overuse or misuse of antibiotics in humans, is also being like misused in other sectors as well. For instance, the agriculture practices and animal farming as well. The farmers tend to overuse antibiotics to increase the production and things like that. They randomly just mix it with the feeds and use it in the farms. Again, this is an issue. So what happens is that all this overuse and misuse of antibiotics, this bacteria is getting exposed. more and more to these antibiotics, and this creates a selection pressure for the bacteria to emerge and become resistant to that particular drug. So that is how antibiotic resistance emerges among the bacteria that's in and around us. Now, other causes of antibiotic is like poor infection control practices in the hospitals and clinics. This tends to cause more infections, and again more antibiotics are being used. So if we are following proper infection control strategies in the hospitals, we can stop infections being coming and then being prescribed with antibiotics. Again, lack of personal hygiene and poor sanitation, and finally lack of new drugs being developed. So these are the major causes of antibiotic resistance. Now, this is just a brief timeline of the antibiotics discovery as well as the development of antibiotic resistance. So in 1928, the first antibiotic, that is penicillin, was discovered by Alexander Fleming. And after that, you can see as the time passes, many new antibiotics comes into the market. But as soon as a new drug comes into the market, we can see within no time, bacteria becomes resistant to the particular drug and it becomes ineffective. So what happens is that it's because of the overuse or misuse, these antibiotics are soon becoming ineffective. So the rate at which a new drug is developed is like it takes years and years for a new drug to come into the market, but the rate at which bacteria develops resistance to that particular drug, it is like just quick as possible. So that shows the issue, seriousness of the issue. And in the present day, we all, individuals, the researchers and the policymakers, we are all fighting together against this antibiotic resistance issue. Next, this map here, it shows the global burden of antibiotic resistant bacterial infections. We can see like each country in the burden varies, and an estimated 1.2 million people, yeah, 1.2 million people died in 2019 alone due to antibiotic resistant bacterial infections. And it is predicted, according to the latest predictions, which was published in Lancet, it states that nearly 40 million people could die by 2050. That is, the death toll due to antibiotic resistant infections is going to rise by 70%. That is like, it's not like fast, just in 25 years that's going to happen, according to the predictions. So this is why we call AMR as a silent pandemic. Because during the COVID-19, we know that the impact was clear, the outcome, like people were dying like around us. So we were all scared. But in the case of AMR, it's just happening like silently, it's emerging, and we are not seeing that on a daily basis. The impact is not visible on a daily basis. So people are not aware, or people just don't care. But that is a serious issue. Now, AMR is a One Health issue. So just like it affects us, it also affects the environment as well as the animals around us. So it's a One Health issue. So today I'll be talking mainly about AMR in the environment. Now, what are the key sources contributing to AMR in the environment? We know there are various like pollutions that's happening around us. So I'll just pinpoint few of the major sources that is contributing to the antimicrobial resistance issue in the environment, which includes the bathing waters and the shellfish harvesting waters, etc. So first one is the municipal wastewater. The community wastewater as well as the wastewater from the hospitals includes the municipal wastewater. So this is like it has a high load of pathogens, which includes all the microbes and high load of antibiotics in them. So this is like a hotspot for emergence of AMR. So that is one of the major key sources of AMR contributing to the environment. And other is like industrial discharges. So what happens is that there are various like pharmaceutical industries which are producing high amount of antibiotics, and they are discharging their like unused antibiotics or their like effluents directly into the environment. So that's another source. And finally, all the treated waste or slurry from these wastewater plants, etc., they get dumped as landfill into our environment, or they are used as manure for crop production and things like that. Then another issue, another contributor is the agriculture runoff, the surface runoff from the agriculture farms, as well as the animal production sectors where antibiotics are used in high loads. So all these surface runoffs and all these discharges finally ends up in the environment. So here I just produced a flow chart of and how these antibiotic resistant bacteria and antibiotics are finally ending up in our environment. Now, what can we do? So we as researchers, what we can do is like we can contribute to our surveillance programs. We can do more research on AMR so this way we can provide evidence base which can inform the policymakers to bring out with necessary interventions. So strengthening surveillance is one of the major things like what we can do to tackle AMR, that's a silent pandemic. So coming to surveillance, choosing the right approach has a major impact. So we, in our lab, we are a wastewater monitoring lab. So we mainly focus on wastewater-based surveillance or wastewater-based epidemiology. So what is the power of wastewater-based surveillance? So I'll just discuss it here. So it is a very powerful, currently we can see like wastewater based surveillance is being used in many areas of various disease tracking systems because it's a most powerful tool because it provides like a comprehensive information about the total health of the community, and it provides a unbiased reflection on the community's health and lifestyle habits, and it can be used as a complementary approach for current infectious disease surveillance systems, and it can act as an early warning system for disease outbreaks for tracking the rise and spread of AMR within the community. It's already been used in various sectors for monitoring poliovirus or SARS-CoV or influenza virus or tracking the use of illicit drugs within the community, etc. So along with this, we can use this approach for monitoring AMR as well within the environment. Now, we know like now we have chosen the right approach, maybe the wastewater based surveillance. Again, choosing the right tool is also very important when it comes to AMR surveillance. So the tool depends on what you actually want to look at. For instance, like if you're just interested in a particular pathogen, let me say E. coli. So you just want to see whether a particular E. coli or the E. coli of interest is antibiotic resistant or not. Then you can just go for traditional culture-based methods, and then you can do the culturing, and then you can test the particular pathogen for antibiotic resistance using the traditional antibiotic susceptibility testing using distribution or broad dilution methods. And this method is used just for culturable pathogens. So according to the report, it states that nearly only like 1% or something of the microbes are culturable in the lab. So this has its own limitations. So we don't have any information about the unculturable bacteria, and also we get no information about the type of antimicrobial resistant genes that is carried by this bacteria. So then when it comes to PCR or polymerase chain reaction, it is more targeted. So if you are interested to look for the presence of a gene, for instance, the colistin drug resistant gene, you want to look for the presence of this gene in your pathogen, you can go for targeted PCR methods. And then you'll get information about the presence of the gene in that pathogen. However, it also has limitations. For instance, you don't get any information about the novel or emerging antimicrobial resistant genes within the community. So again, so both these methods has its own limitations. However, if you're interested in just looking at a particular aspect of AMR, you can use this. However, if you want to you want to get a comprehensive information about the whole community, microbial community within the sample of your interest, then you should choose metagenomics because like it is non-targeted. So you're not missing anything. You're getting everything that's hidden within the sample. It gives us a comprehensive information about the total antimicrobial resistant load or total antimicrobial resistance genes that is present within the sample, and it profiles the entire microbial communities. You also get an information about the resistant mechanisms that is prevalent in your sample on your community, and we can track the emerging variants, and we can also link the antimicrobial resistant genes to its bacterial host. And it also gives us information about the horizontal gene transfer via mobile genetic elements. So overall, metagenomics is a powerful tool for community level surveillance. Now here I'll share a few case studies showcasing the genomic driven AMR surveillance which we have conducted in our lab here at Bangor University. As I already mentioned, we have a wastewater monitoring lab, National Wastewater Monitoring Lab. So we mainly use a wastewater-based epidemiology for our surveillance program. And we use two type of tools, that is high-throughput qPCR as well as metagenomics in our surveillance. So we mainly go for high-throughput qPCR to look for targeted genes of our interest. So if when we are interested only in looking at particular antimicrobial resistant genes, that is genes that confers resistance to a particular drug, then we go for high throughput qPCR because we have a pre-selected non-AMR genes which we are interested to look at. We use this, and it is rapid and it has a faster turnaround time as well. And then we choose the metagenomics approach for the untargeted shotgun sequencing. And here we get information both about known and potentially novel AMR genes. And we can also link the AMR genes to the potential host by using various binning tools and things like that. So this gives us an comprehensive information about the whole community. However, it takes longer time. The processing time is long, and it includes bioinformatics as well. I'll just briefly explain the wastewater sampling and processing we do here for AMR. So we have refrigerated auto samplers which are installed at our hospitals and wastewater treatment plant sites. So what happens is that drivers go out to these hospitals and wastewater treatment plants, and we collect 24-hour wastewater composite samples which we bring to our lab, and we centrifuge them and take the pellet, and this pellet is further used for AMR analysis. So after the pellet is taken, we extract the DNA, and depending on what our interest is, we either do metagenomic sequencing for comprehensive surveillance, or we go for high throughput quantitative qPCR for targeted monitoring. So the first example, we have used metagenomics-based approach. So the study is already published. So if you're interested to read further, you can get the information from the paper. So in this study, what we did was we collected 24-hour wastewater samples for 1 year, that is from January to December 2023. And we collected the hospital wastewater, untreated hospital wastewater, which was directly from the hospital outfall pipe. We collected the hospital wastewater, and then we collected the influent that was entering the wastewater treatment plant, which includes both hospital as well as the community wastewater. We also collected the effluents, that is the treated effluents coming out of the wastewater treatment plants. And then we were analyzing all these samples together. What we found was, so these are the box plots which shows the total AMR abundance which was found in hospitals, wastewater treatment plant influents as well as effluents. And we could see that there was a higher load. You can clearly see this is the blue one is the hospital effluents. The load of AMR genes, that is the burden of antimicrobial resistance was as expected, was much higher in the hospitals compared to the community wastewater. When the untreated influent as well as the treated effluent was compared, we could see that there was a reduction. After the treatment, there was a reduction in the total load of AMR in the effluents. However, none of the treatment plants under our study was efficient enough to remove all the antimicrobial resistance from the treated effluents. This shows that the current wastewater treatment methods are not effective enough or they are not targeted to remove antimicrobial resistance from the water that is finally entering our environment. So this way, a baseload of AMR antimicrobial resistant genes and antimicrobial resistant bacteria are still being released continuously into the environment, which is used for various recreational purposes like bathing or for kayaking, or we use these waters for various shellfish or other harvesting purposes as well. So there's a high need that the current treatment method, we should start inventing, or we should start using advanced treatment methods like quaternary level treatments or other advanced oxidation methods, which can potentially remove more of these pollutants, which includes both the antimicrobial resistant genes, the antibiotics, as well as the antibiotic resistant bacteria from the effluents in the wastewater treatment plants. Sorry, again, the diversity. The diversity means the type of antimicrobial resistant genes that is present within a sample. So again, the diversity of the genes were also higher in the hospitals compared to the wastewater treatment plants. However, you can again see there was still a lot of these genes present in the treated effluents as well. Now, this is the stack bar plot. Here we can see that the different type of antimicrobial resistant genes that were present in the wastewater samples, which we studied for a period of 1 year. And the pattern remained like almost similar throughout the year. That means like it is endemic and these genes are present in the samples throughout the study area. And we can get the information of the type of antimicrobial resistant genes that were present in the hospitals as well as the influence and effluents, and we can see the pattern like varies slightly. For instance, here the blue color refers to genes conferring resistance to sulfonamide antibiotic, and the yellow refers to tetracycline antibiotics. So these genes, the abundance of these genes were much higher in the hospitals compared to the community wastewater treatment plant. But however, when it comes to wastewater treatment plant, they had a slightly different pattern. For instance, the rifampicin resistance, that is the rifampicin is the antibiotic which is used for treatment of TB, Mycobacterium tuberculosis. So it was much higher in the wastewater treatment plants compared to the hospitals. So this is the kind of information which we get when we use metagenomics. We can get the information on the total type of antimicrobial resistant genes that is present in the sample. So based on this, we can like advise the hospital authorities or the public health authorities. So see, we are seeing a high resistance of tetracycline or sulfonamide in your hospitals. So you should improve your antibiotic stewardship practices, and maybe you should try to um, stop or reduce the usage of these drugs and then use like much effective drugs instead of these drugs here. So that's the kind of like suggestions we can put to the public health authorities using our research data. Now, this is a network diagram. It's based on a prediction. So this network diagram shows the connection between the bacteria and the different antibiotic resistant genes. So higher the size of the node means higher is the connection. And the purple color refers to the bacterial genera and the green color refers to the antimicrobial resistant gene. And you can see the connection between these. And many pathogens such as E. coli and Pseudomonas and Klebsiella had high connection with many clinically relevant antibiotic resistant genes. So this type of method or approach can predict the specific bacterial species or genera that are likely to carry certain antimicrobial resistant genes within our sample. This is highly useful for pinpointing the bacterial reservoirs of AMR genes and hotspots, guiding targeted interventions. And we can like advise the infection control team in the hospital. See, this particular pathogen, for instance E. coli, is carrying high load of AMR genes in that, so you should target like disinfection approaches targeting this particular bacterium. Now, in our second study, we used high-throughput qPCR. So this service was done at Resistomap, Finland. So they had like a chip. It's like a chip, a panel or small chip in which we can test up to 384 AMR genes at a time. And we were using a 96 gene panel, and using that we tested wastewater samples as well as the receiving environments, the wastewater receiving environments as well. So we collected again the hospital wastewater as well as the community wastewater as well as the treated effluents were collected along with the receiving water, sediments as well as the shellfish samples from the receiving environments as well. Now from our results, we can see that the patterns of antimicrobial resistance that was prevalent in the hospital wastewater treatment plants, the receiving coastal water and sediment. And the abundance was much higher in the hospitals, and also it was like totally unexpectedly we found that in the coastal sediments also the relative abundance was much higher, and the pattern was similar, the composition was similar with the wastewater samples. So that means that wastewater effluents or the treatment plants have an influence on the receiving environmental waters and sediments. And here we can see we quantified that using qPCR or quantitative qPCR, we can actually quantify the load of AMR genes that is present in the sample, which is not possible with metagenomics, because metagenomics is a qualitative approach where we can only study the relative abundance of things. However, if you're interested in quantifying the load that is present in your sample, I would suggest using the quantitative qPCR approaches. And here, when we quantified, we could again find that the load of AMR genes were much higher in the hospitals then influence. However, the load was similar to the influence and the sediment. So sediment in the environment, the environmental sediments were acting as a reservoir. We would suggest like they are acting as a potential reservoir for antimicrobial resistant genes. So during resuspension of water, during a high storm event, the sediments get resuspended with water. Finally, all these AMR genes, which are being like hiding in the sediment, which will end up in the water and which we'll get finally exposed to. Now we did a Venn diagram to see how many of the AMR genes were shared within these different type of samples, and we could see that out of 96, 71 of the antimicrobial resistant genes were shared between hospital, effluent, the environmental sediment and water. That means it's all shared. So all these, what we are seeing in the environment is it will be potentially coming out from the wastewater from the hospitals as well as the wastewater treatment plants. Now we again try to estimate the removal efficiency of the wastewater treatment plants. We were interested how effective the treatment method was to remove the total bacterial load as well as the total AMR load from the treated effluents. So here the green refers to the influent and blue refers to the effluent. We could see that, and we studied it from like January to June. We also wanted to see whether there was a seasonal difference in the removal efficiency of the treatment plants. And we could find that there was a seasonal variation in the removal efficiency. And though throughout the study period, there was a reduction, you can see. However, a high load was still being discharged. This is gene copies per liter. So a high load of AMR genes were still being discharged to the treated effluents. Again, when we looked at the bacterial load, that is 16 sRNA gene copies per liter, we could again see, though there was a reduction, a high load of bacteria, potentially antibiotic resistant bacteria, were being released to the treated effluents. This means the treatment method is not efficient. The current treatment methods used are not efficient in removing reducing the burden of AMR to the environment. Now, our next experiment, we tried to see how much of AMR is being accumulated in shellfish samples near the wastewater outfall pipes. So what we did was we collected fresh oyster and mussel samples from a local farmer, and we put them in a cage and we deployed them near the outfall pipe of our wastewater treatment plant. And we deployed it there for a period of like 1 week. So after 1 week, we took it back and we tested them. So before deploying, we collected few fresh samples. We tested them for AMR, And then after deployment for a week, we took them back and we again tested them for AMR. And we could see that post-deployment, the relative abundance of antimicrobial resistant genes in the oysters as well as mucil samples increased. And we could also see clinically relevant genes like colistin resistant genes in the deployed effluent. So that means these sewage outfall pipes, there are lots of shellfish harvesting areas in and around the sewage outfall areas in the environmental waters. So they are being continuously exposed to these effluents, and through which all the shellfish are taking, and shellfish we know they are filter feeders, so they take up everything. So they're filtering everything. So it finally ends up in their flesh. And if we are eating half cooked or like the raw shellfish, we are taking up everything like through food consumption. Finally it ends up in us, like, so we'll be taking up using the shellfish sample. So this shows the seriousness of the issue and how bad the impact of the sewage outfall is on our food chain as well. Now, what can we do to this? So we can definitely tackle AMR. So the global action of AMR was launched in 2015 by WHO in collaboration with with FAO and World Organization of Animal Health, and they had 5 major strategies to combat the issue of AMR, which includes like improving awareness by raising awareness among the community about AMR through proper effective communication, education and training, and then strengthening our knowledge and providing evidence base through surveillance and research, then reducing the incidences of infection by following proper hygiene practices or infection control measures, and then optimizing the use of antibiotics, that is preventing the use or overuse or misuse of antibiotics, and using only antibiotics only when it's needed. That's very important. Use antibiotics only when it's needed, and then invest more on development of new medicines or new antibiotics or new alternative therapies for antibiotics as well. And what can we do to tackle AMR? We definitely have a role and we can do something to tackle AMR as well. So we includes government, researchers, the healthcare professionals and the policy makers. So by strengthening our surveillance system, by doing more research on AMR, we are providing more evidence to the government and the policy makers. See, we have an issue here. We have high load of AMR in these water bodies which we are using on a regular basis. So the environment authorities or the public health authorities should do something. See, we have the evidence, so you should do something to tackle this. So we as a researcher, we can provide that evidence to our government and policymakers. And then what the government can do, they can like invest more on research related to finding more development of new antibiotics, so using like more antibiotic alternatives. Like, so we have alternatives to antibiotics. For instance, bacteriophages. Bacteriophages are viruses which feed on bacteria, so they kill bacteria. So these and they don't infect us, so we are totally safe from bacteriophages. They don't infect us, though they are viruses. They only infect or they kill only the bacteria. So we can use bacteriophages, for instance, in the aquaculture practices and all, that can be used as an alternative to antibiotic. This way we can reduce the usage of antibiotics, or using like vaccination programs, or like more vaccinations can reduce the infections. This way we can reduce the usage of antibiotics. And in the hospitals, they can follow proper antibiotic stewardship practices, using or means reducing the usage of like the last resort drugs and using more of the other available drugs. And this way they can minimize an overuse, misuse and overuse of the antibiotics. And then mandating treatment of effluents before it's been released into the environments. So there are cases where untreated sewage during a high storm event or things like that, all this untreated sewage have been directly disposed or released on a regular basis to the environment. So the government can bring out like strict monitoring or monitoring of these discharges, and they can enforce strict regulations so as to control these discharges. This way we can reduce the pollution of our environments. And then overall, a multi-sectoral collaboration is definitely needed. So we have to do something. We have to provide the evidence, and the government should use that evidence to bring out necessary policies. And the environmental agencies should also like have a strong monitoring program so as they monitor when there is no improper or unregulated discharge of waste into the environment. Now, we can definitely do that because I have seen few success stories, which I'll just share here two of the success stories of tackling AMR. First one, according to the latest reports from UK government, we could see that in UK, in the agriculture practices, we have made a like significant reduction in the antibiotic usage and the AMR. So this plot here, it shows the percentage of E. coli, fully susceptible E. coli. That means E. coli that is sensitive to antibiotics. So the percentage of E. coli that is sensitive to antibiotics has increased over the years. At the same time, the percentage of E. coli that is multi-drug resistant, that means which are resistant to multiple drugs, it has decreased over the years. It's only because you can see in parallel the antibiotic sale and the usage has reduced over the years in the agriculture practices. So when there was a reduction in the usage of antibiotics in the agriculture or the animal production sector, we could clearly see a reduction in the emergence of antimicrobial resistance as well. So this shows that if we reduce the usage of antibiotics, that is being reflected in the emergence of AMR. The emergence of AMR is also like simultaneously coming down. In the other example, the Norway's fishing industry is always cited as a good example how a sector should be run. Because in Norway, they cut down the usage of antibiotics in their salmon farms to virtually zero. So how they achieved this was using by vaccinating salmon and using bacteriophage therapy as alternatives to antibiotics. So this led to reduction in the antibiotic usage, and the WHO also recognized Norway for this effective effort that they took in to control AMR. So this shows that we are still not like the time has not run out. We can still do something to save the remaining antibiotics for future generations. So I'd like to conclude by quoting the saying by Sir Alexander Fleming, the inventor of the world's first antibiotic, that is penicillin. Sir Fleming had rightly predicted that we are going to have an issue of antibiotic resistance as soon as he invented penicillin, because this is the quote he said: the thoughtless person playing with the penicillin treatment is morally responsible for the death of the man who succumbs to infection with penicillin resistant organism. So that shows that he had rightly predicted, and now we are seeing that the issue of AMR. So we should all, I would suggest we should all take one step at a time, whatever we can, though it's a small effort, we should all put in our efforts to combat the global issue of AMR and to save the remaining antibiotics for our future generations so that there'll not be any treatment failures or more deaths in the future. Thank you so much for listening.
Thank you, thank you, thank you so much for that excellent presentation. We really appreciate you. So now it's time for questions and answers. So I will open the floor for you. If you have questions, you can ask directly, or you could also type it on the chat, whichever one is convenient for you. If you have maybe any concepts that was talked about that you really don't understand, you want more elaboration on, please feel free to ask.
Like if you need more information on the methodology we used and things like that, you can just like read the papers. Maybe I can share the link to the papers to the organizers so they can share that so people get more information on the methods that we used and things like that in detail.
So I have some questions and I'm really excited about this. Okay, I think there's a question on the chat. I don't know if it's Oh yeah, so someone is asking that you should elaborate on bacteriophages therapy.
Bacteriophages therapy. So as I already mentioned, bacteriophages, they are viruses which can kill bacteria. So just like an antibiotic works, so how does an antibiotic work? It kills the bacteria so that it no longer infectious. So the similar way, the action of bacteriophages is also like it kills the bacteria that is causing an infection. So instead of using antibiotics, you can use like bacteriophages. So bacteriophages now come like as cocktails and things like that. So the patient that it is now currently being used for treatment of many infections where antibiotics are no longer effective. So there were certain instances like a patient was I think like I don't remember exactly from where it was. So none of the drugs was working for that particular patient, and so the doctors had lost like complete hope of treatment of that infection. So that is when the patient's family from someone they heard about this bacteriophage therapy, and they contacted these people who were like producing this bacteriophages, this company, and they imported that. And when the patient was treated with that bacteriophage, that is virus that killed that was supposed to kill that particular bacteria, it was effective and we could save that patient. So that's what like bacteriophage therapy means, use of viruses to kill bacteria instead of using antibiotics.
Okay, thank you. And someone else is asking that what is the best treatment method for a wastewater treatment plant before releasing effluent?
Yeah, currently, like there are many new approaches like advanced oxidation methods, which are proven to be more effective in removing antimicrobial resistant bacteria and genes, which are currently like invented by people around the world. So it's like a quaternary level of treatment. So this can be used or combined treatment can be also used UV plus advanced oxidation. So like that combined treatment can also be much effective. I can't hear you. Sorry, you're muted.
Oh, sorry, can you hear me now?
Yeah.
Okay, Victor is saying nice presentation, thank you. I wish to ask if when doing the gene screening, you can predict from which organism the resistant gene came from.
Hmm.
Maybe like during the metagenomics process, like can you like accurately predict what organism the
For
resistant gene?
metagenomics you're asking, right?
Yeah, so he's asking about the gene screening. That can you
Yeah.
predict from which
So what I showed was a network-based prediction to show from which organism that a particular gene came. However, instead of that, you can also use like we are also using the binning approaches. So what we do is like from the metagenome dataset, we assemble the genome and we create the whole bacterial genome. And after binning, using various binning tools like MegaHit and MetaBat and MaxBin, we produce the whole bacterial genome from the short-read metagenome dataset. So once the bacterial genome is generated, we again look for and screen for antimicrobial resistance within the genome to find what this bacteria is carrying. So that way we can um, not read it, we can actually point out which is the host bacteria that is carrying these genes.
Okay. Okay, thank you for that. Shitu, do you want to ask a question? Please go ahead. Yeah, your hand is raised. You can go ahead, Shitu. Okay, you are currently muted in case you don't know.
Okay. Hello, ma.
Yeah, we can hear you.
All right.
Hi.
Good afternoon. Thank you very much, ma, for the presentation. My second question is is about the disposal of the antibiotics. I got to understand that it also has a link to the escalation of resistance. So most people, what they tend to do is burn them or dispose them in the toilet and even when they are burnt, they can still cause resistance. We had of a research which was done on some antibiotics that were burnt by the agency in Nigeria, the NAFDAC. They burnt some falsified antibiotics, and the sites, the bacteria they got from there, they were resistant to multiple drugs due to the exposure they had to those antibiotics. So what is the best way to dispose maybe falsified or unused antibiotics, maybe in the hospitals or outside the hospitals? So what is the best way to dispose them? Thank you very much.
That's a tricky question because, like, I don't know what's the best way. There are various ways. The first thing is like, see, this we have lots of antibiotics, and each has a different degradation rate. Few antibiotics, as soon as they are released, I mean, or discharged or disposed, they tend to degrade. But few, for instance, beta-lactams, they degrade faster in the environment. However, there are certain antibiotics like, for instance, vancomycin, that tends to remain in the environment for a longer time. And so they have a longer degradation time. So if you ask me, like, what's the best practice? Of course, you should never, like, put down the antibiotic down the drain. That's not suggested. You should never do that, even in the hospitals. We know that the nurses sometimes they do that, but that's not advisable. So within the hospitals now, there are technologies like there are new, just like I said, like advanced oxidation methods are there for removing AMR bacteria. Many new methods have come up, which I don't remember which all, but there are definitely new methods which have been invented which can degrade these antibiotics. So collect all these unused antibiotics and then degrade them, or then before treat them before it's been released, so that it doesn't cause any more like selection pressure on the bacteria. So like don't discharge them just like that down the drain. That's not advisable. But other methods should be used. But if you ask me like what's the best method, I don't have an answer, sorry.
Yeah, and yeah, just to add to that, I know in the hospitals they do have incineration where they have this, like it's a very big oven like place where they burn all hospital waste. So most times they also destroy the antibiotic waste inside the incinerator, and it's such that it doesn't end up in the environment. Yeah, because it's always done at a super high temperature, so it doesn't end up in the environment. But one of the best rule is don't ever flush your antibiotics into the toilet, because it ends up into either the septic tank or into the waste water treatment facility, and then all of this thing that Dr. Reshman explained then occurs. Yeah. Then also in some developed countries they have this antibiotics take back program where you go the antibiotics that you didn't finish using, or maybe an excess antibiotics, back to the pharmacist where you got it from, and then they have a proper way of disposing it. And for instance, the issue you highlighted about NAFDAC, I'm sure one of the things that happened is after they burnt the antibiotics maybe in an open site, then it's there was like a runoff of these burnt antibiotics into maybe a nearby river body or maybe environment. It led to the resistance of AMR in this bacteria. Now, thank you very much. We still have some questions in the chat, and I think we have like 5 more minutes left. So Juma, okay, I think Idaya asked the question. She said, can antimicrobial resistant bacteria in treated effluent adapt to different environmental conditions?
Of course they can. Of course.
Okay.
Bacteria, see, that's the main like advantage of bacteria. In order to survive an environment, they tend to like emerge and evolve rapidly, so that they can like, just like they describe selection pressure of antibiotic tends them to become antibiotic resistance under different environmental conditions. Also, they tend to evolve so that they can survive. It's like survival of the fittest, the same strategy.
Okay. Then Henry is also asking that you mentioned horizontal gene transfer in the environment. how significant is this pathway for spreading resistance compared to direct human or animal transmission?
We did find many horizontal gene transfer genes like integrons and mobile genetic elements, mobile genetic elements, what was it, like transposons and things like that in the wastewater samples. So wastewater are like a hotspot or reservoir for all these mobile genetic elements. So it provides that it is rich in nutrients, it has the bacteria, it has the antibiotics, and all the mobile genetic elements is there. So it's the right ambient condition for horizontal gene transfer to occur. And activated sludge methods in the wastewater treatment plant, the sludge treatment level, that stage is known to be like promote horizontal gene transfer. And we could see that when we are comparing the influent and effluent bacteria, the community were different. So there was definitely some horizontal gene transfer that occurred between the environmental bacteria and the clinically relevant bacteria. It's very important. Even in the environment is also known to promote horizontal gene transfer.
Yes. So, Henry, it's a very significant pathway also, just like the direct human and animal transmission. So, Mayowa is also asking the question. He says, thank you for your presentation. For metagenomics, how do we further identify the novel AMR genes?
Novel AMR genes. Further identify the novel AMR genes. I have to look into that.
Yeah.
We have found many like novel bacteria,
Hmm.
uncultured and novel bacteria from a metagenome dataset. So in similar way, there should be one approach. There should be some approach. We haven't looked for novel genes in our metagenome dataset, but there should be some method for sure.
Then I also wanted to ask, like, does the sequencing depth, like, the depth of sequencing and
Yeah.
all that, does it help in identifying maybe the low abundance
Mm.
genes that could be novel?
Yeah. I means when you increase the abundance, the low abundance genes tend to come in, and our depth was like very high, like 150 to 250
Okay.
billion base pair per sample. So we choose a like higher depth sequencing. So depth of the sequencing is also very important. That's how we found out the novel bacteria also, like the hidden reservoirs from our samples, which were carrying AMR genes.
Okay,
The
thank you.
that's called like the microbial dark matter. We could find many carrying these AMR genes.
Okay, thank you very much. So Juma has a question. She said, are there any specific timelines or milestones associated with the global action plan that you talked about?
Each country has a national action plan. For instance, UK, it has now come out with its latest action plan, that is 2024 to 2029. So it's like 5-year action plans. So each 5 year, that particular country, so according to the global action plan, each country should also have a national action plan to implement. And so each 5 year, they look how the action plan worked and how much effectively they could reduce AMR. Then from that, the next 5-year action plan is being designed. So it's like 5-year action plans.
Yeah, that's true. Because even in Nigeria, we recently launched the National Action Plan, the second phase last year. And I think it's usually for around 5-year
Yeah.
period.
So
Yeah.
then we'll see how the progress is, whether there was any progress, whether there was a decline, or there was actually some reduction, and based on that, new plans are being launched.
Yeah, thank you. Then I think you've answered this question that Anna is also asking. She's asking that, are there other ways to treat effluent disposal to reduce antimicrobial present in the waste?
Yeah, that's like advanced treatment methods. As I already mentioned, we do have treatment methods that's invented by many researchers, but the water companies, they should like in our country, like in our area, I would say now I say we are now talking with the water companies, and we are estimating the cost of implementing an advanced level of treatment in the wastewater treatment plants in the North Wales, UK. So we are now exploring different methods and we should have a cost-effective method as well. Few methods are like very costly and expensive, so the water companies may not agree with that. So, and cost-effective and the most effective method. So
Yeah.
according to that, we should be implementing advanced. Of course, quaternary level of treatment is definitely needed. And we should have a pre-treatment at the hospital sites also. Pre-treatment of hospital wastewater is definitely important because that hospital wastewater is the major contributor. So if we can reduce the AMR at that source, at the hospital source, that still reduces the burden that is entering the environment. So pre-treatment of hospital wastewater is definitely important.
Okay. Yeah, so I actually have like some questions from my hand, but I'm like, oh, the time is up. But I know you focus majorly on hospital wastewater and all that, but also poultry farmers, livestock farmers, they also use antibiotics on their farm, and then it gets discharged in the environment and all of that. So do you look into the agricultural sector to look at the wastewater around that sector, and what, if so, what are the results that you've been observing?
So as I already mentioned, in UK, I mean, the veterinary medicine departments have been like very strongly advocating reducing the usage of antibiotic, and so
Mhm.
in the animal production sector, there has been a rapid decrease in the usage and rapid decrease in the AMR as well compared to other sectors. That's
Oh,
why we are mainly focusing on the hospital sector because in the animal production sector, there's definitely a reduction, which we are seeing over the years.
okay. So how about in like developing countries like Africa, Nigeria, where we don't even have a standardized wastewater treatment plant, for instance. Most people just maybe septic tank, or they just dispose their waste directly to the environment. What is the best method for monitoring AMR in those kind of environment?
I have to think about that. Because now we are trying to monitor AMR in Barbados.
Okay.
So they were having an AMR issue there. So again, they were saying they don't have a proper discharge of wastewater and things like that. So we are collecting wastewater from the animal abattoirs and the aircraft. So we can see, like using aircraft monitoring, we can see whether people coming to the country is carrying AMR from other regions. And then you can directly go to the hospital and then from the collect wastewater from the ward level surveillance or things like that you can do. So you can find like what's the status of AMR within the hospitals and within the community. I think you should like do grab sampling of the septic
Mm.
tanks and then use the composite and then test that. If there's no proper treatment plan, grab sampling of the septic tanks and then taking the composite and test would be good.